It was good. Today we got circulatory system. So yeah. So what does the heart provide? It provides the force or the pressure for blood to kind of move through the body. This helps with blood circulation. Outer layer of the heart, inner layer of the heart, three subs and purpose. So the outer layer is called the I don't even remember this one. Pericardium. Pericardium. That's the protective layer, and then the inner layer is called the myocardium. And endocardium lining. Okay, I need to look at that one. Anyways, what is the upper and lower parts of the heart called? So the upper parts are called the atriums, the atrium chambers, and then the lower parts are called the ventricle chambers. So the right side and the left side are different in that the right side has deoxygenated blood and it'll pump that to the lungs, while the left side has the oxygenated blood which will pump to the rest of the body. What is the pressure difference between the two chambers? Why? So the atrium, those are less pressure. They have less blood pressure because they're just like taking blood from the veins, right? So it's not really too much. But the lower chambers, they're getting blood forced into them, especially the left ventricular valve chamber. That one's like the thickest one. It has like the most muscle. It gotta pump blood all the way to the rest of the body. So that's why it's pretty thick. What are the three valves? So first on the left side, or on the right side, on the deoxygenated side, we have the tricuspid valve. That's the first one, and then afterwards we have the pulmonary valve. Then we have the bicuspid valve, which is also called the mitral valve, and then we have the aortic valve, which is the last one that pumps that, which was the last one before pumping it like to the rest of the body. So what is important about them? It prevents backflow of blood. That's like the main thing they do. So what does the aorta do? It's just a place for the left ventricle to pump oxygenated blood into and then distribute it to the rest of the arteries to the body. That was it. Okay, so how many chambers do fish, amphibians, reptiles, okay, so fish have 2 valves, amphibians have 3, reptiles also have 3, and then birds and mammals have 4. The 2 exceptions to reptiles are crocodiles and alligators. So it's just like crocodilian. Yeah. So what's the order of the valves that the blood goes through? So blood's going to come in deoxygenated from the rest of the tissues through the superior and inferior vena cava. It'll go into the right atrium, and then it'll go through the bicuspid valve, or no, the tricuspid valve. It'll go into the right ventricle. Then it'll go through the pulmonary valve to the lungs. After the pulmonary valve, you're going to come back from the lungs into the left aorta. After that left aorta, you're going to go to the bicuspid or mitral valve. Then you're going to go to the aortic valve to the rest of the body. Okay, so what are the difference between arteries and veins? So arteries bring blood away from the heart to the rest of the body, or like just away from the heart, and then veins bring blood back towards the heart. We're talking about differences in, okay, we'll talk about that later. So the systemic and pulmonary circuits, pathway from heart. So systemic is like to the rest of the body, so it's from oxygenated blood towards the rest of the tissues, then bringing deoxygenated blood with the veins. Now, pulmonary circuits are just going from bringing deoxygenated blood from the heart to the lungs, and then back, bringing oxygenated blood back to the heart. So, so difference in blood concentration, highest and lowest. So the highest oxygen concentration will be in the pulmonary veins, because they're bringing back oxygenated blood that's been freshly oxygenated, while the least oxygenated blood will be like the superior inferior vena cava, because their veins bringing back the blood that's been used all over the body. The difference between arteries and veins. So I think for this one I'm talking about like the special difference between pulmonary and systemic. So pulmonary arteries will bring deoxygenated blood to the lungs, while systemic arteries will bring oxygenated blood towards the rest of the body. And then the opposite is true for veins. So veins, the pulmonary veins will bring oxygenated blood from the lungs to the heart, while systemic veins will bring oxygenated blood deoxygenated blood from the rest of the body to the heart. Inferior and superior vena cava, they're just, the top and bottom. They bring they bring the blood to the right atrium. This is like kind of all the veins combined, kind of like just like into one big group, closer to the heart. So 2 phases of the cardiac cycle: systole and diastole. So I just put that there. So systole and diastole. Systole is when like depolarization, I think it is. Okay, systole is contraction. Diastole is relaxation. What would happen if pacemakers were absent? So pacemakers are what create the stimulus for the heart to beat. And if that were to happen, the heart would not be able to beat properly. It would not be good. I think that's all I really have to say. Yeah, no synchronized heartbeat. How are gap junctions important for the heart? So without gap junctions, the stimulus, or like the electrical signal, will not be able to move in a coordinated way throughout the heart. So pathway of conduction in the heart. So first we're going to start with the SA, the sinoatrial node. Then we're going to go to the AV. I don't know what that stands for. AV, ventricle. Okay, I don't even know. Then we're going to go to the bundle of His, which is like kind of the septum in between the right and left ventricle, and then the Purkinje fibers will distribute the rest of the stimulus to the ventricles to pump them. What is cardiac output? How do you find it? So cardiac output is the amount of blood pumped per minute, like the volume of blood, I think. Stroke volume multiplied by heart rate. So stroke volume is the amount of blood pumped like in one pump, and then heart rate is just beats per minute. So you could find how much blood is pumped per minute through cardiac output. How do you find stroke volume? It's the diastolic versus systolic pressure. So, that matches systole and diastole. So you're going to look at the amount of blood when the blood is rest, like when the heart is at rest, so when it's like filled, and then like before contraction, and then systole, you're going to check the volume after the heart contracts and sends the blood out. How do you find blood pressure? CO and CVR. So I know for sure CVR means, systemic vascular resistance. So this is the resistance for blood to push through. There's 2 factors. It's like the viscosity of the blood, or the narrowness or radius of the blood vessels. CO, cardiac output. So how much blood is getting pumped into the body, and then what is the resistance of that blood throughout the body, kind of. So how do arteries and veins differ in thickness? Why? So arteries are more thicker, I would say. They have more muscle. They have to pump blood to the rest of the body. Veins, they have less pressure. The main difference, because like the main reason they have a difference in thickness is because arteries are under higher pressure, so they have that muscular tissue around it to like prevent it from bursting and just be stronger. While veins, they are less thick because they have less pressure, but they still have muscle, So so arteries thicker for withstanding high pressure. Veins have valves. So I don't know if we talk about, but veins have I think I made something like, what's special about capillaries? But they have valves in there, so they like don't like go backwards because it's like less pressure. They also use like the force of smooth muscle around them to help get pushed out to the superior and inferior vena cava. How many layers of cell in capillaries? Okay, what's the primary factor that influences blood pressure? It's the diameter. Vessel diameter. It's the systemic vascular resistance due to cell diameter. So capillaries, how many layers of cell in the capillaries? I believe it would just be one layer of cell so that they could do diffusion effectively. So how many layers of cell in capillary? There would only be one layer of cells so that the well, actually, it will be simple squamous cells, so that it helps with diffusion between the cells and the blood. There you go, connecting. What is the smallest diameter vessel but greatest total surface area and cross-section area in the body? Capillaries. What do capillary beds do for us? Wait, what are capillary beds? Oh, so I really don't remember. Okay, so they regulate exchange, they filter, and they do nutrient delivery. I feel like it was different in the video, but I will perhaps look into that. What is the difference between oncotic and hydrostatic pressure? So hydrostatic pressure is the pressure put on like within the blood vessel. So like it's like the proteins and stuff inside the blood vessel, which will push out the nutrients and gases that and the oxygen that are meant to go out of the blood vessel. While oncotic pressure is the pressure from outside of the cell, trying to push in like the wastes and things like that, I believe. Yeah, so hydrostatic pushes fluid out, oncotic pulls fluid in. What do precapillary sphincters do? So these are responsible for, they're like, so they can control like the blood flow. So if you're cold, it'll like restrict the blood flow. It's called vasoconstriction. Then there's vasodilation, which is when you're warm, and it'll like decide how much blood to pump, like where blood should go, like how much blood should be going to different places. 4 layers to the vein. What do they have special about them? Oh, I didn't even talk about why 4 parts of the artery. So the innermost is going to be the endothelial cells, or will it be the outer? Yeah, I think the innermost. I want to say the innermost is the endothelial cells. Let me ask this thing. Okay. So they have the inner layer composed of endothelial cells, simple squamous epithelium. Tunica media, the middle layer, smooth muscle cells and elastic fibers. Outer layer, connective tissue. So they have 3 layers. There was a 4th layer I remember in the video, but okay, well, I guess I'll just look into that later. But the thing they have special about them, okay, I don't know, but okay, so the inner layer is the endothelial, then they have the smooth muscle layer, and then they'll have the connective tissue on the outside. The special things about veins, like I said, they have valves that prevent blood from backwards flow, and then they also use the smooth muscle contractions to pump blood, to just help with that. How does a fetus get and remove blood? Through the placenta. So it'll get blood through the pulmonary artery, pulmonary vein. So it'll bring oxygenated blood from the mom's placenta towards it, and then it'll give deoxygenated blood through the placental vein to the mom placenta. So what does the ductus venosus do? Okay, so I really don't remember the names, but I just remember the order. So ductus venosus is responsible for bypassing the liver, because the liver is non-functional in the baby, the fetus. So why would it give blood to the non-functional organelle? So it'll be like, psych, you're not getting no blood, and then it's just going to skip that and then move on. The foramen ovale, that connects the right atrium and the left atrium. It doesn't, because usually it's like deoxygenated blood and oxygenated blood, but that oxygenated blood that's coming in, it's like, it doesn't need that, or wait, hold on, I'm getting confused. Right. It connects the two, because in order to get to the left atrium, you have to go through the pulmonary system, right? You're going to the lungs, but you don't need to go to the lungs because you don't have no freaking lungs, right? So it's just a way to connect the two, so you don't waste time passing through the lungs. And then the ductus arteriosus thing, that's like a secondary backup to the foramen ovale. That'll connect the pulmonary artery, right? Pulmonary artery to the left atrium. Let me just make sure. Oh, to the aorta. What's the aorta? The aorta is the artery that takes away deoxygenated blood, right? So, or oxygenated blood. Yeah, okay, I think so. Okay. So what kind of system is required for blood pressure? A closed system is required for blood pressure. A closed circulatory system is required for blood pressure. Like if you just have blood flowing around, you can't have blood pressure, you know, like otherwise you're gonna burst. So you gotta have veins and like stuff like that to make it have blood pressure. What can high blood pressure lead to? You know, organ damage, things and such. Not very good. Yeah, so organ damage, vein damage, artery damage, everything damage. It's just not good. Heart strain as well. So the main counter is called, like I said earlier, vasodilation. Vasodilation will dilate the blood vessels and the capillaries, and then it'll be like become wider so that you have less SVR. I really don't remember what it stands for. Systemic ventricle. I don't even resistance or something. Okay, anyways. Yeah, so what does low blood PSI do? Well, you might not be pumping enough blood to your brain, and that's not good because your blood needs brain. So what is the main way to deal with low? It's the complete opposite. It's called vasoconstriction. So vasoconstriction will tighten up the blood cells, and then it'll be harder to pump. So what are the two regulatory processes called? I think vasoconstriction, vasodilation, I think I just said. So yeah, what are two other ways to regulate blood PSI? So kidneys. So when the kidneys scrub the blood, they can control how much urine or how much water gets pulled out of the blood. So if you pull more blood, then you're gonna have lower volume, then you're gonna have lower pressure. If you pull out more water, then you're gonna have lower volume and lower pressure. Or wait, did I say that twice? I don't even know, but it's like the same way, vice versa. The other way to regulate it is, I think it was like increased temperature control. What? I swear it was not temperature control. I'm like, bro, okay, wait, let's put a star on that. Okay, where is blood pressure the highest and the lowest? Highest would be the aorta, lowest would be the superior and inferior vena cava. Yup. Okay, what are the three distinct layers that come from centrifugation? Okay, what is blood what is blood characterized as? It's connective tissue, because it's suspended in plasma. What are the three distinct layers that come from centrifugation of blood? So first, the most dense is the red blood cells, or just the cells. You can have the red blood cells, the erythrocytes, the leukocytes, which are the white blood cells, and you can have platelets. Next, you're gonna have the buffer, which is the buffer coat. It's just, oh wait, no, sorry, I was wrong. I was completely wrong, bro. I was wrong. The most dense, I was kind of originally right. The most dense is erythrocytes. That's going to be the red blood cells. Second dense will be platelets and the white blood cells, leukocytes, or lymphocytes. But now I'm getting confused, bro. I think it's leukocytes, right? But I gotta pee, bro. Okay, wait, where was I now? And then that's going to be the buffer coat, and then the least dense will be the plasma, which will just sit on the top. What is the main function of red blood cells, white blood cells, and platelets? So red blood cells are mainly used for oxygen transport, maybe even CO2 transport sometimes when it feels like it. White blood cells are used for immune response, and then platelets are used for clotting. That makes sense, right? Yep. Okay, bro, there's so much more. I gotta pee. Okay, what do antibodies do? What are antigens? So antibodies are things in the blood, in the white blood cells, or like just in the blood plasma, that look for antigens. Antigens are things that help identify what a cell is, if it's foreign or if it's part of the body. So which of these determine blood type? Antigens will determine blood type. Antigens will be present on the red blood cells, and then you'll be able to detect foreign or not. So four types of types. Which antigens? Four types of type bloods, I think. So there's A, B, and then there's AB, and then there's O. So antigen on A will be A, antigen on B will be B, antigen on AB will be AB, and antigen O, there's nothing like the antigen O. Blood type O has no antigens. So now this brings us to the question, which antibodies does each one have? So in a human body with antigen A, they're going to have B antibodies. With a human body with antigen B, they're going to have A antibodies. And then blood type O, they have no antigens, so they're going to have anti-A and anti-B antibodies. And so what is the reaction of foreign blood types? It's called agglutination. What does it lead to? It leads to clots, and it's just not good because it's really dangerous. So if you that's why blood transfer is really important to know which type is which. What is the rhesus factor? Rhesus puffs, eat 'em up, beat 'em up. Rhesus factor is positive or negative. So if you have the rhesus factor, you're going to be positive. If you don't, then you're going to be negative. So the true universal donor is going to be O positive. Wait. O negative. O positive. Hold on. No, O negative. Because if you're O negative, then you have no antigen. So then you're able to like chill in, right? Yeah, you have to be O negative. Right? Yeah, okay. And then the universal recipient would be AB positive. Because if they don't have the antigen, so they don't have anti-A, they don't have anti-B, they also don't have anti-RH. Because they have the rhesus thing, so then it's going to be like, what are they going to be targeting if there's only two, right? So that's why. So O negative will be the true universal donor because it has no RH factor, nor does it have any A or B stuff. So yeah, what are the two benefits to the biconcave shape of erythrocytes? So the first one, so it's red blood cells. So two benefits: well, increased surface area because you get more surface area to carry more oxygen, and the second one probably like smoother maybe. Flow, flexibility. Oh, so I guess to fit more. So yeah, flexibility, smoothness, etc. Okay. Do red blood cells have organelles? No, they do not, because that organelle would waste space for carrying oxygen. How are prosthetic groups attached? Covalently bonded. Right? Yeah. Okay. So what is the minority of CO2 get transferred? A minority gets transferred through the red blood cells. A majority of it is transferred as bicarbonate ions in the blood. Right? Yeah. Yes, okay. How does myoglobin do things? It's just a storage area for oxygen when muscles are working. Where is bicarbonate found? What? Okay, so it says bicarbonate buffers blood CO2 levels. Okay, so that's another function. Don't really know where bicarbonate just found anywhere, probably like blood anywhere, like wherever it feels like being, probably. What is another function of bicarbonate? It's a buffer. What is carbonic anhydrase? It's like it catalyzes the reaction between bicarbonate to water and CO2. Yeah. So what is the major buffering systems inside cells? So inside cells, intracellular, that is phosphate. Outside the cells would be bicarbonate, or carbon, or whatever, right? So what is blood-brain barrier? It's like a highly selective membrane that coats like the blood vessels that go into the brain, and just barely allows anything to go through. It just allows like fat-soluble vitamins, and then steroid hormones, and what else is there, like oxygen, and etc. So what is the purpose of clotting? The clotting is like to prevent excess loss of blood when you bleed. Like if there's an injury to the vein, it prevents that. So the steps of this, I don't fully remember this, so I'm going to put a star there. But what usually happens is, let me just think about it. So you cut your hand, right? You cut your hand, now the vein's broken. So platelets are going to escape. Those platelets are going to call something. What are they going to call? Fibrin? No. There's like a trypsin or something in there, right? But wait, not trypsin. It's something, bro. Something. It's something in there. Let me just look at the notes if they will decide to load. Bro, I need to pee, bro. Can this load, bro? Can this load? Page 6. Where's the... Bro, there's... Damn, why did I look at a lot of stuff? Okay, so first we're going to have the enzymatic cascade. So the platelets are going to go get the enzymatic cascade. Prothrombin will come in and it'll get activated to thrombin from the enzymatic cascade. Now thrombin is a positive reinforcer. Wait, no, not positive. I don't even know, bro. Positive like activator or something, and it'll positive reinforcement. No, I don't even know. But it'll like cause more enzymatic cascade, more thrombin to be produced. Now that thrombin is going to go from cause fibrinogen to get into fibrin, and the fibrin is like fibrous proteins that will like cause the blood to clot and hold everything together. So yeah, that's all. I'll see you later for what even chemical kinetics.